afternoon, dear students. Last time we discussed about strain development. So let us continue with that. So what is a strain? Now see, why strain development is important or strain improvement is important, because once we have isolated the organism producing a desired product by primary and secondary screening, it becomes necessary to increase the product yield so that we minimize the production cost. And how can we increase the product yield? The most important approach is improving the productivity of the strain. So any technique or approach that genetically modifies the strain to increase the production yield is known as strain development. Okay, what are the different methods of strain development? Mutant selection, recombination, and recombinant DNA technology. Last time we discussed about mutant selection. Now let us continue with the second method of strain improvement, that is recombination. What is recombination? It is the formation of new gene combinations among those present in different strains. Genes from two different organisms are combined in one. Okay, so that the newly constructed recombinant gene has desirable characteristics. Okay, so once several different mutants have been isolated, recombination can be used for genetic analysis and strain improvement. So we can use, once you have isolated the organism, you can use recombination for genetic analysis as well as for strain improvement. Okay, the desirable characteristics, the desirable alleles present in two or more strains can be combined into one to increase the product yield or to generate new products. Okay, so desirable features from two or more organisms are combined in one so that we get a good yield or we get new products. How can we do that? Recombination can be achieved by transformation or transduction or conjugation. Okay, that is called as sexual cycles or parasexual cycle and protoplast fusion. Okay, what is transformation? When a foreign DNA is absorbed and integrated with the genome of the donor cell, that means a foreign DNA, a naked fragment of foreign DNA enters inside the recipient cell. The recipient cell accepts a fragment of foreign DNA and the foreign DNA gets integrated inside the DNA of recipient cell. And hence the recipient cell becomes a recombinant cell because its DNA becomes recombinant. Okay, a foreign DNA gets integrated, enters inside the recipient cell, and the foreign DNA fragment gets integrated into the DNA of the recipient cell. Thus we get a recombinant cell. But in order that the cell accepts the foreign DNA, it has to be competent. Okay, and competent is not a permanent phenomena in the life cycle of an organism. So cells can be deliberately made competent by treating or by growing them in calcium chloride. This method has been used to increase the level of protease and amylase production by bacillus species. Okay, so transformation was used to increase the protease and amylase enzyme production by bacillus species, and this method has good industrial application. Coming to conjugation, conjugation is a process of recombination in which there is a contact between donor and recipient cell, and through a pilus which we call as sex pili. Okay, it's a hollow tube which passes from donor to recipient, and there is transfer of plasmid from donor to the recipient. Okay, and this is a unidirectional transfer. It goes from donor cell to recipient cell. Okay, so this type of conjugation has been the mycelial conjugation takes place among actinomycetes with DNA transfer, as in the case of eubacteria. Okay, so conjugation is a means by which we can develop recombinant cells, right? The third is transduction. What is transduction? It is transfer of bacterial DNA from one cell to another cell by bacteriophages. Okay, so bacteriophages, that means bacterial viruses. So bacteriophages are the vectors which carry genes from one cell and transfer it to another cell. So the second cell becomes a recombinant cell which receives DNA from other cell. Okay, in this process, phage attaches to the donor cell, takes by mistake picks up the DNA of the donor cell, lyses the donor cell, and the donor DNA is injected into the second recipient, that is host, and the second cell now becomes a recombinant cell. Transduction is brought about by two processes, is of two types: generalized transduction and specialized transduction. In generalized transduction, any part of the gene of the donor is carried by the phage to the recipient cell. And in specialized transduction, a specific gene loci like gal gene or biotin gene is transmitted from donor to the recipient cell by the lambda phages. So whether it is transformation or transduction or conjugation, these are the three modes of recombination forming recombinant genes. Okay. The next method of recombination is parasexual cycle. There are certain industrially important fungi which have asexual mode of reproduction, right? But sometimes the hyphae having haploid nuclei, hyphae of hyphae of the asexual fungi having haploid nuclei fuse to produce a heterokaryon. Okay. Sometimes most industrially important fungi are asexual. However, their haploid hyphae often fuse to produce heterokaryon. Heterokaryon means a cell having nuclei from two different hyphae. Okay, two different nuclei are present in the same cell. Now, sometimes the two nuclei of the heterokaryon fuse and form a diploid nuclei. Okay, and after mitotic division, the diploid nuclei breaks up into haploid nuclei, giving rise to recombinant cells. Okay, this type of phenomena is helpful in developing recombinant fungi. Okay, so parasexuality is a rare form of sexual reproduction which occurs in some fungi. Okay, in this case, the hyphae from different strains, hyphae having haploid nuclei from different strains fuse and form heterokaryon. Okay, this type of phenomena is important in those fungi like Penicillium, Chrysogenum, Aspergillus niger, wherein no sexual reproduction takes place. Okay, so developing recombinants with the help of parasexual cycle is important in fungi which are industrially important having asexual mode of reproduction, right? Now the next method is protoplast fusion. Okay, what is a protoplast? A cell without a cell wall is called as protoplast. So a cell is specifically grown in presence of certain enzymes, or it is treated with certain enzymes like lysozyme, or it may be treated with chemicals like polyethylene glycol so that it loses its cell wall. So when a cell without cell wall is called as a protoplast. In case of protoplast, the outermost membrane is the cytoplasmic membrane. Okay, so protoplasts are formed from bacteria, fungi, yeast, actinomycetes. When dividing, cells lose their cell wall. How can they lose their cell wall? By treating them with enzymes like lysozyme or by treating them with certain chemicals. Okay, now protoplast fusion enables recombination in strains without efficient means of conjugation. Okay, in case of protoplast fusion, protoplast, that is cell without cell wall of two sexually incompatible plants or protoplast of two different bacteria can be fused. Okay, so the protoplast of two different organisms is taken. They are allowed to undergo fusion. The fusion is brought about by treating them with certain chemicals like polyethylene glycol or sodium nitrate in presence of sucrose, cytochalazine B. There are different chemicals, but the most widely used chemical for fusing the protoplast, two different protoplast, is polyethylene glycol. Or if we can also give electric shock. Okay, so that the protoplast of two different organisms fuse, and when the protoplast fuse, the contents that are present inside the cell also fuse. Sometimes the two nuclei from the two protoplasts may fuse Okay, otherwise they do not fuse. When the nuclei from the two protoplasts do not fuse, one nuclei is specifically lost, what we get is a cytoplasmic hybrid which is called as a hybrid. Okay, so protoplast fusion is also a means of developing new cells, right? So protoplast fusion has been successfully done with Bacillus subtilis and Bacillus megaterium and several species of Streptomyces. Okay, okay. It has also been done between the fungi, right? Geotrichum and Aspergillus. So protoplast of these two organisms are fused and we get a new hybrid cell having desirable characteristics, right? This type of method is helpful in developing new strains which give high yield of antibiotics, especially okay, high antibiotic yielding organisms. High antibiotic yielding strains can be obtained by this method of protoplast fusion. Now the third technique for strain improvement is recombinant DNA technology, which is also known as genetic engineering. What it involves? Cut and paste technology. We isolate a desired gene from one organism, put this desired gene into a cloning organism, a vector like a plasmid or a cosmid or a phasmid or a phage. Okay, so the desired gene is isolated, it is spliced or inserted into a vector, and then this newly formed recombinant vector, recombinant plasmid, is inserted into a cloning organism or a host organism, and the gene, the foreign gene that is inserted into the vector is allowed to express in the host organism, and we get the desired protein product in the host organism. Okay, where do we use this technique? For producing recombinant proteins and for modification of metabolic pattern of organism to produce new product, modified product, or more quantity of product. Okay, so recombinant DNA technology involves isolating a gene of interest from one organism, putting it into a vector, and then inserting or allowing the expression of gene in the host organism, right? And the technique is used for production of recombinant proteins and modification of metabolic pattern of organism, so that we get a new product or more yield of the product. Recombinant proteins like insulin, interferon, certain recombinant vaccines can be produced by using bacteria using the transgene, and all these products have commercial importance. Metabolic engineering. In case of metabolic engineering, the metabolic pathways of an organism are manipulated. Okay, the metabolic characteristic, the metabolic pathway of the organism is modified or manipulated in such a way that we get maximum product yield from the foreign gene that is inserted into the recombinant organism. Okay, so metabolic engineering is a process in which a process of designing and redesigning of metabolic pathways of an organism, wherein we modify the metabolic pattern, the metabolic pathway of the organism, such that we get maximum production of the desired product. The existing pathway in the wild type organisms are modified. Okay, they are slightly modified, or entirely new pathways are introduced into the organism through genetic manipulation, through the manipulation of genes, so that the improved yield of product is obtained, undesirable side products are eliminated, or entirely new product can be obtained. Okay, so the existing pathways, metabolic pathways in an organism are modified, or completely new pathways are introduced by introducing a foreign gene inside it, such that you get more yield of the product and you reduce or eliminate the unwanted products or side products, or you to produce a totally new product by using metabolic engineering. Metabolic engineering has been used for developing strains which give you more antibiotic production or a different type of product formation. Say for example, This type of technique is used for overproduction of amino acid isoleucine in Corynebacterium glutamicum. Ethanol by E. coli. As such, E. coli doesn't produce ethanol, but with the help of metabolic engineering, we can make E. coli produce biopolymers like bioplastics, PHBs. We can make E. coli produce ethanol by introducing a foreign gene inside it. So overproduction of antibiotics, production of ethanol by E. coli, production of PHB, which is used for manufacturing a biopolymer in E. coli. Okay, then modifying organism such that we get a totally new product from the organism, or we get more product yield from the organism. That is called as metabolic engineering. Okay, so that is all about strain improvement. Next time we will discuss about sterilization of air, sterilization of media and all.